We can see you. So hello everyone. We're getting our presenter ready. Hey, yes, we can hear you. Welcome. Hello.
Hello. We were fighting with the tech to get in.
It looks it's working now. Fantastic. So welcome to our webinar. There are already people joined. I think you started the meeting by pressing the start, but let's try to share the slides if possible.
Okay, I can share the slides.
So while you start getting started, I'll start with the introduction. So welcome to our 39th webinar.
Okay, yes. So yeah, we are still trying to get something.
Okay, it looks promising. Yes. So the general rules of the webinar are here that attendees are muted and video off until end of the presentation. And well, we kind of agree that the talk is supposed to be about 45 minutes. Is it okay like that?
Yeah.
Perfect. Yeah. Fantastic. Thank you very much. Then, well, we started already recording. I also should mention that this webinar is supported by the IEEE, Engineering in Medicine and Biology chapter of Czechoslovakia section of IEEE, and also by the International Union of Radio Science, URSI. So welcome everyone. Today is my great pleasure to introduce a really giant in the field of terahertz spectroscopy, Professor Martina Havenith. She's a physical chemistry professor at the Ruhr University Bochum in Germany. And she's also spokesperson of the Cluster of Excellence Ruhr Explores Solvation and the director of the Center of Molecular Spectroscopy and Simulation of Solvent Control Processes. And as many of you might know, she has developed and pioneered new infrared and terahertz laser technologies to explore fundamental questions in chemistry. Particularly, her research area is in solvation science, and she currently also explores and develops terahertz calorimetry, linear and nonlinear terahertz spectroscopic methods, and the role of water in fundamental biological processes such as formation of protein condensates. But without further ado, Martina, I would like to give word to you and accompany us through your talk. So please go ahead.
Great. So before I start, I want to thank you. I want to also thank the Cluster of Excellence Resolve, which is one out of two clusters of excellence in chemistry in Germany, which I'm directing, and also the ERC, because I will talk about Terahertz calorimetry, which was a granted ERC advanced grant. So it was high risk, and I have to say it didn't work how I proposed, but it worked, and it works even better than I thought before. So, but this is science, right? So I want to start with stating that Watrix is the matrix of life. So we know when researchers search for water at other planets, they always search for water because they think that is so far one of the ingredients which is needed for life. And if you think about this, so also we have like 75, 80%, 85% of water in all our cells. So depending on how young we are. And so we talk a lot about biologists, people talk a lot about proteins and so on, but most of this is water. And also biology is putting a focus on water. And you see that here, and this is what end 2023. So where they were studying two what they call different form of water, and I will replace this. These are like schematic sketches of what they think water is in cells, and I want to give you during this talk a different, more distinct, pronounced view on what that means. These are just paintings. So they talk about that the solvent thermodynamics is very important for cell death and for preventing cell death. And they talk about something like bulk water and structured water. I have to say there's no bulk water in cell because it's really crowded inside there. But this is just what their sketches, and I think we can put a better picture on this. So we also started in 2015 with saying that water, there's more than this, the role of water in the cell. And there's like being originally a physicist, so the easy view was also with this other water and the other water. But what does it mean? You see here that if you have a substrate and enzyme on molecular binding, you might have different types of water at different times of the substrate, different sites. And when you bind something, when you bind like a spaghetti to a substrate, it's more fixed. And in this way, so what you have is that it's entropically unfavored to bind because you lose a certain number of configurational space. And then we found out that there are preferential binding sites which are there where the water is a little bit different than the other water, where the water is already entropically unfavorable in the binding site. And I want to give you, when I teach entropy and water, I always give the following example of entropic cost. So what you all know is if you look in a catalog, Pitt's room looks like this, right? So this is like the very organized state with one configurational space. But then if by time, or especially if you have a kid's birthday, so then the whole thing will look like this, which means there are a lot of configurations which can be taken, right? Each toy can be at a different position. And usually if you do it statistically, then each of these configurations will appear. But in order to get it like in an organized state, you have to pay what is called entropic cost. So somebody has to pay the entropic cost. Everybody who is a parent knows who is paying the entropic cost after this. But it means that thermodynamically, so by itself, it will go in a less ordered state. So if you have let it be like this. So what means that here in this regime, it will not binding to a substrate is entropically unfavored. It's more, it's one state and it's not many states, whereas before it can fluctuate. So it's entropically unfavorable, so it has to be compensated by binding additional forces or interaction. And usually in biology, this the difference of this is very close to zero. So we have delta H, which means with favorable binding interactions are there, and T delta S, so that you want to go to to the less organized state, and both of them are nearly in equilibrium. So it's free energy delta H minus T delta S. And if you are close to zero, then in biology you can change things. If you are like in a very fixed state, you cannot change them anymore. So you want to be close to equilibrium. Biology doesn't want to be in equilibrium because then you are dead if you're in complete equilibrium. So you want to be able to tune it within several degrees. as you do actually if you become fever. You tune with 2 degrees. And in this way, we are very close to this, and both of them are important, delta H and T delta S. There are a lot of methods which are very good in delta H, also spectroscopy methods, where you look for binding, NMR, EPR, fluorescence correlation, and so on. But T delta S is very hard to get, but it's equally important. And therefore, so I proposed, okay, there is calorimetry, a well-known technique in biology, where you measure delta S and delta H also by putting something in a sample, heating it up, and measuring the heat transfer. So you measure the heat capacity and the change. But if you do this, you need a certain sample volume, a macroscopic one, and transferring heat is slow. So you can be in the millisecond regime. I mean, usually our second regime, if you want to measure heat transfer. And temperature is always macroscopic. So the idea was, can we connect spectroscopy with calorimetry? Because if we can connect spectroscopy with calorimetry, then when you can do this, you can do all the tricks of spectroscopy. You can do nonlinear spectroscopy. You can do time resolved spectroscopy with picosecond. You can focus, and you can also do beyond the lateral resolution limit spectroscopy. So then you come into a new way of doing calorimetry. And so we propose, we will explore how, if we can make a connection between calorimetry and spectroscopy, which sounds strange. But terahertz is in the frequency range of the populated state at low frequency. So we said, let's see if we can connect it. And so I will now do this and show what do we have, what modes we have in the terahertz, and maybe this is what is known more. So if we go to the low frequency regime here, we have the hydrogen bond stretch of water, and we have a libration. So what is this? Every water molecules will break hydrogen bonds and open on a time scale of 1 picosecond. If you do the reciprocal of 1 picosecond, it's 1 terahertz. This is the regime of terahertz. And there are 2 major modes. One is the intermolecular stretch, which I have sketched here, where they go away from each other, and that probes something like the hydrogen bond strength, the radial part of the interaction. The other part at higher frequency is when one water molecule rotates in the water network of the other one. So the other ones are like connecting it, having an interaction. That's why it's usually we have this tetrahedral hydrogen bond network. And if you rotate it, it's a hindered rotation, and that will be in the range between 600 and 700 wave number. And that probes more the rotational or angular part of the potential energy surface. And so what do we know? How is calorimetry defined? There are 2 components to calorimetry. One is, which is known, if you put a solute into water, the first thing is you have to create an empty cavity before the solute can be put in. So the water around the solute here will form a cavity around it. The other thing is that if you insert a solute, you insert it like you do with salts, for example, if you have a positive interaction between the water and the solute. So there are 2 terms in a Gedanken experiment. Of course, you will put it at once, but in a Gedanken experiment, you can dissect it in 2 parts. One is forming the solute, a water around a solute, and the other one inserting the solute. And we put it here. You see these sketches here, how you form it. Here, the water connects with itself, and here you also make hydrophilic interaction with the solute. Let's see how these show up in the spectrum. And the first thing for like forming these cages is what is called class rate. Class rate are like a buckyball of water forming around it. Inside is something hydrophobic, so it doesn't want to bind with the water. And then you form a class rate around these guests. So this here is in form of a buckyball, so you can find them. Class rates are found deep in the sea at low temperatures there, where they are, where they form these stable cages, but it's not an ice crystal cage, because in the middle there's this hole here. So, and around this, the water from a net. When we looked at these spectroscopically, and I say this is the Bavarian form of it, so since the water cannot connect with the solute because it's energetically unable to connect, they put up their two hands or their two hydrogen bonds. One they put to the next neighbor here, and the other one they put, like what is here, the legs, they put outside to the bulk. So one will find the partner at the side, the other one will go outside. So they have no bond with the inside, which will be important, unlike in ice. And if you look at this, so you can see below here, you have the spectrum of water, bulk water. There's 194 wave numbers, is the center of this hydrogen bond stretch. And then ice here is at 210 wave numbers. So you see the ice, this is just normal frozen ice, not well structured, is blue shifted, the center peak frequency compared to the bulk. Why is that? Because if you are physicists or chemists, you know, omega is the square root of force constant divided by the reduced mass. So if you have the force constant is increased as it is in ice is very stable, then the force constant is higher, so the frequency is higher because the reduced mass is that of water. Now, when we looked at this class rate, we found another peak at 165 wave numbers. So it's lower than that of bulk water. And lower means a lower force constant. So it's weaker bound than bulk water and much weaker than ice. And why is this? Because now we have something, and we call that a wrap, a net. We have these hydrogen bonds around the solute, preventing the solute, and this is more two-dimensional than three-dimensional. So it's missing cooperativity. Ice is very stable because it's very cooperative. The four all sticks the other water and keep it in the middle. Bulk water is opening and closing, but still on average tetrahedral coordinated. This one here has only two coordination partners, so it's weaker bounded, and that's why there is this initial band here. This is the class rate band at 165 wave number, redshifted. So you can say, okay, good for class rate. So, but what is in general? So when we think of thermodynamics, forming a cavity around it, and like putting here, this is an alcohol with a hydrophobic side, and here one hydrophilic, one hydrophobic, there is this 164 band, which we see when we measure the difference of a spectrum with the alcohol inside and without the alcohol. So this cavity formation thing here, this cavity wrapped water around it, forming the cavity, you can predict, this is a red prediction, will have a band at 164 wave numbers, similar like this cleft wave. This is the signature of this part in the thermodynamic cycle. And you could see here, we measured it. So you see, these are Fourier transform measurement. We measure the sample with the solid in blue. We have the reference spectrum bulk of water. You do density correction, and therefore you subtract the missing bulk volume. And when you do this, you find this is the difference for ethanol, for butanol. And you see here a maximum in delta alpha, so the difference to bulk at 164. So this cavity formation spectrum is there. It's temperature dependent, and it depends how long the hydrophobic chain is. The longer the hydrophobic chain, so butanol is longer than ethanol, the higher the maximum is. So this is like, can we use this cavity formation? And we said, okay, let's try these amplitudes, which you see here with the orange arrows, if we can connect them to this part of the thermodynamics. So what we did is, by the way, do you see the full picture?
Yes, we do. It's perfect.
Okay. So what we plotted for the alcohols delta alpha. So this is the orange arrow, how big this is, against the well-known delta S. So the changes in entropy, because we said this cavity formation is the one which determines the change in entropy. So the more we have around it as the cavity, so the more cavity we have around it, this is a loss of configurational space, because any cavity is a loss of possible configurations, and this determines delta S. This was a poor theoretical Gedanken experiment. But now we said, let's connect it with spectroscopic observables. And you can get this delta S. I mean, you can put alcohol and water in usual calorimetries. And then you see here, we plotted this against delta alpha, and you see a perfect linear correlation. It was, I think, much better than what I thought. It really works. So you get them all on one. And when you plot them, this means you can now fit this correlation factor, this linear one, between delta alpha and delta S. So you get the change in delta S from the spectroscopy. So we have now done the first part. Creating the cavity here in this thing, we can determine, and we get delta S when we put a solute in from measuring delta alpha. We have a scaling factor which we get out of the fit, and this can be determined. Now we have to do the second part of the thermodynamic cycle, putting a solute inside. And what does it mean? This is more hydrophobic, and this is hydrophilic. Hydrophilic means we form a hydrogen bond, and this determines delta H, so the enthalpy. And so let's go for this one. So how do we get this? We discussed the 164 band, but we see that all alcohols at all temperatures have here, if you plot the difference to bulk water, there's a negative delta alpha, so which means smaller absorption or smaller population, around 300 to 400 wave numbers, and more between 500 and 600 compared to bulk water. So this is only when you do the difference. And when you think is, what do we have there? we have here the librational band spherical hindrance. We have the one, this is, you can put it in, it's inhomogeneous, so there are softer librations. So the one which can still the water, it's an ensemble, rotate freely. And then there's the other one which is stiffer. And stiffer hydrogen bonded, so it cannot rotate so good because it's more bound than to the other waters by the solute. And this means the force constant is higher. This means this frequency get more rapid, the frequency shift. So we have more of these high frequency, less of the low frequency. And I'm sorry, this is terahertz, this is wave numbers, but it would be more here, less here. So this is the sign of hydrophilic bound water. More at 600 wave numbers, less at 300 wave numbers. And this we tried, is that correlated with delta H? Because entropy is determined by water molecules, solvation entropy by water molecules which have more interaction with the solute than bulk water. And so we did that, and you could see all alcohols have the same signature, which makes sense because all alcohols have one OH. So all of them have the same hydrophilic part. And it's also less temperature dependent because if you have a stronger bound, it's less temperature dependent than if you have a weak intermolecular interaction. So this was a hint. All alcohols have the same, that this is due to the one OH. And when we did this, but we wanted to also get like a reference, and maybe you focus here on this side. So what we said is, we will look for the slope. We cannot determine the frequency shift with our spectrometer because it's out of reach of our frequency range. But we have less here, we have more here. And this determines the slope, how steep this is. So we did take the slope and plotted it against delta H, but not for the alcohols because all alcohols have the same slope. So we did do that for glycerol water solution. So for something which has a lot of hydrophilic bonds. When you do that, we see that scales linear with the slope. So we have a correlation factor between how steep this is in the delta alpha and how delta H mixing is. This has been measured by traditional calorimetry, so you can look it up and you see it's nicely linear correlated. So now we have our two cycles in thermodynamics put and correlated with spectroscopy. Creating a cavity here, we see the 164 band, and then inserting a solute, so and getting delta H, we do by the slope. We have the scaling factor, so for alcohols, we are done. Now you can say, okay, for alcohols, now you did a lot of things to do this, but we want to apply the same model for very different applications, where we cannot do the easy calorimetry. And I will show you this here. So in general, I have to say, so people thought the Oscar in biology should always go to the protein. These are the only ones who determine what we do. But I want to show with measuring the thermodynamics that the water is not just the crowd here. This is Kant. These are the actors on the red carpet. We want to investigate how much determines water, which is like always around it, like the crowd, determines biology. What is the role of the water? And we want to do that now to something more complex. So neurotoxic aggregate formation, liquid-liquid phase separation. This is now a different thing. You have a few quantities and there it's very inhomogeneous. Now you want to understand the role of the water in this process. Why? Because people are wondering how these aggregates are formed, they see that it's formed like spontaneously protein clumps. They are still liquid. They are not yet the fibrils, which everybody knows of in Alzheimer and so, which are irreversible. But before they form protein condensates, highly concentrated protein. And of course, if you think of that, this would be like in your cup of water, all the sugar which is dissolved would go to one clump of sugar. This does not happen spontaneously for sugar in your cup of coffee, right? It's not formed. But there have to be two things. So the protein-protein interaction must be favorable because entropically it's not favorable to go that all proteins are in one side, so which are marked here in green, and that the rest is empty of the proteins. As I say, think of your coffee cup. The sugar is equally distributed. This is entropically normal. Now they go all together. So it must be favorable, but unfavorable entropically, but it must be by protein-protein interaction favorable. But what can you maybe, what is the role of the water in that game? Because it's tunable. Again, you can see it's very easy to tune with co-solutes, temperature, and so on, this process. So it's nearly again in equilibrium. If the protein-protein interaction are very strong, proteins form just a fibril, that's it. That's the end. No more change from there. But usually they go in this one. And we wanted to investigate what the role of water is. So when you do this, you go to your medical people in the nearby department, you ask them for, we did FAS, which is very much in respect to these neurotoxic aggregates investigated. And what they can do is, so you put the protein in solution, you see this here on the left side, and then you can trigger protein condensation by putting something inside which cuts one side of the protein, and then they spontaneously form droplets. This will be like when you mix oil and water for your salad sauce, and if you wait, so the oil will form a droplet in the water. It's still liquid, it's not solid, but it will be a phase separation. So then you wait because the droplets are heavier, have a higher density than the bulk solution. So with the time, all the formed droplets will be on the surface. And what we used was a attenuated reflection crystal. So where we go in from the bottom here with evanescent wave, and it probes everything on the surface, of course, in the terahertz, like in the micrometer range. And what you do is, so these sink down, you see them here in the fluorescence picture, or here you see the cover glass protein droplets on the spectrometer. So with the time, you will have more and more droplets, which means in the beginning, you measure the dilute protein solution. In the end, you measure mostly the droplets. So then what we do is, instead of measuring the difference to bulk water, we plotted the difference of delta alpha between the start when it was diluted and the end when the only droplets were on the crystal. So it's the difference between the protein condensate and the dilute protein solution. If you plot that in the terahertz, you find something really interesting. We didn't know before what we would find, but first thing, what you see with time, you see that the slope here, you recognize the slope. So you see that the slope is increasing in delta alpha, which means you have more and more hydrogen bond water on top of the crystal. The number of hydrogen bond waters contributing to delta H is increasing with time. And this is because in the protein condensate, the proteins are highly concentrated. They have hydrophilic water on their side. And if you increase the protein condensation, you also increase the number of hydrogen bond water because they go with the protein. So this you see very clearly here. Now, I said there's a 164 band, and what we could see is for the hydrophobic water around the alcohols, it was positive here. Here, it's getting more and more negative, which means we lose the hydrophobic water. When we form protein condensate, so the hydrophilic water is increasing, so this is retained in the protein condensates. Why this cavity wrap or this hydrophobic water is released in the bulk. And you see both signatures very nicely, the 164 and this here. What you don't see is the protein. I always say, if you want to look at the protein, go to the infrared. The protein may be like this little dimple here. It's very small. But it's not, there's much more water there, and it highly absorbs in this frequency range. What you see is the changes in the water populations. But you can quantify them, how much this change, how much this change. And then you remember we could correlate the delta alpha with the change in delta S. We can correlate the slope with the change in delta H. Now we do calorimetry upon formation of protein condensates. We use the same scaling factors as for the alcohol and glycerol, and we see how it works. So what is, again, putting a spotlight on this is summarizing. So in the dilute mixture, you have the red, which is, we have the red water, which is the round hydrophobic patches, and this is entropically unfavorable because it has less conformational space. It cannot go, it's fixed to this two-dimensional structure. This is entropically unfavorable. And then we have the blue, the hydrogen bond water, which is enthalpically favorable. So when they do the protein condensation, which is entropically unfavorable, because the proteins, to put them all on one spot, is not favored, as you can see in the sugar water space, but it's compensating this entropic unfavorable situation by getting rid of the water molecules, so the red water molecules, which are also entropically unfavorable. Because if the red water go in the bulks, they can take again all configurations. So there you gain entropy, and you compensate for the entropic loss by putting the protein together. So whereas the blue water, this is the hydrophilic ones, enthalpically stabilizing, this interaction is favorable. So you retain them, and even when you have a more crowded protein, it might be even more hindered, more enthalpically, sterically favorable. So what happens is the water is tuning this formation. You release entropically unfavorable water and retain the other water. And when you retain the bond water, it's still liquid because you still have a layer of water around it. If you would also lose all water, when you lose all water molecules in the bulk, this is a solid fiber. So then you are done. But this here is quite around zero in the free energy. And we could, so what you do is, so you quantify it. This here gives delta S, T delta S. This here gives delta H. And we see that bound water is retained, droplets stay liquids. This is favorable. Entropic driving force also, you release the entropically unfavorable water. So both are contributing to spontaneously driving protein condensation. These are two terms which are not protein-protein interactions, it's protein-water interaction. And you can quantify it. You can see it, you can measure it, and you can quantify it. And this was really exciting that we could do this. So we also, now you say, is this true? I mean, you take this delta H and delta S from alcohols and glycerol. Is that possible? Well, so what we did here is, for this example, first of all, we did see it's very general. On the left-hand side, you see another protein. This was Fas, what we investigated first. This is alpha-elastine. Doesn't matter how these exactly look like. Both do the same thing. They have a dilute phase, and then the droplets form sink down here on the crystal, here, which is this triangle, and you measure the droplets. And in both, you see that this here, they're lost here, and the slope forming here. It's a different amount of both how much you go down delta alpha and the slope, but both do it. And we did find that anyone which forms a liquid phase separated droplets has these two signatures. Very easy. And then you plotted, we said, okay, we can determine delta S here. And we did also, our theoretician calculated delta S, because you cannot measure this. So they calculated here with the model, and then we plotted our experimental delta S using the same scaling factor as for alcohol, so really crude approximation. And also, you know, theory does this, in order to calculate a protein condensate, you also do approximation. But you can see this is nicely correlated. So one is a factor of 2/3 smaller than the other one, but the order of magnitude is perfect. You see that it's linear, and this is the only way to determine it. And both methods, theory and our, have inherent approximation, but it works. And so we were very proud to see that you can do that for this complicated case here.
Do you mind if I jump in just to understand the graph on the right better? So what does the ABCD stands for on the graph on the right? Maybe I missed that. It's the different temperatures. Okay, I see.
Yeah, these are the different temperatures.
Okay, I see. And it's the same protein.
It's the same protein. It's the same protein. Yeah.
Okay.
Oh, there is, I think you see something, there's a picture in between, right? I cannot.
It's okay, it's only, you see it on your side. In our side, we see the whole screen perfectly without any covering, don't worry.
Okay, yeah.
Okay, that's clear to me, thanks.
So these are, we tuned the temperature, and then you can see that the protein condensates forms and goes away, and they also could simulate different temperatures in the system and corresponding to different concentrations. It was known how much concentration you have. You can also measure it with fluorescence, and they simulated different protein concentrations, but doing the same scenario. And also what you can see is, oh, now I'm see if I can, yeah. So what we see is we have here, and I'm summarizing, protein-protein interaction. This is always favorable. Otherwise, if you have not favorable protein-protein interaction, you will never have a protein condensate, right? So you need something to trigger this. So this will lead, but this will be very temperature independent. So this will be because you need a very strong protein-protein interaction to start the process. So this will be always favorable. And it will be delta H, which is favorable, protein-protein interaction. But now you have the other one, but the total thing matters, delta H minus T delta S. Protein-protein interaction delta H small, very strong, and there is entropically, of course, it's not likely that the proteins all end up in one droplet. So now you have the water, and what you do is basically you have the hydrophobic around it and the hydrophilic. The hydrophobic is here now done in red, the other one is in blue. And what you do is you release during the it's a movie, but the movie doesn't, it goes, okay. While you form the protein condensate, so you would release the red one, and you will release it in the bulk. And when it's in the bulk, it's entropically favorable to be in the bulk for the water, because if they have to prevent the solute, this is entropically unfavorable. It's a smaller entropy. They want to increase the entropy, and they can have much more configuration if they are released in the bulk. So you have the protein-water interaction. You use the T delta S is favorable for them to release this type of water. And so that's what it's doing here. So if you put the spotlight on this, during this protein condensation, they will release this water. They will keep the hydrophilic water. So this is how they go here on the left-hand side in the protein condensate. If they release even the hydrophilic water, they are no longer liquid, and then they form a solid aggregate. But the interesting thing was both is important for forming protein condensate. Also to have in the beginning hydrophobic water, because that's the one which is offsetting the entropic cost of this protein condensation. People did not know why a lot of them have these intrinsically disordered chains, why they have these hydrophobic side chains. And you can see that they are important. If you cut the hydrophobic side chain, and then sometimes you are just at the edge where you cannot form any more protein condensate. So both is important to have in the beginning the hydrophobic hydration, so you can get rid of it during the process, and to have the hydrophilic one you need to keep the whole thing in the liquid state. And you can nicely see it. So as I said, we did several, and we could plot a phase diagram of both water populations. And we found that only the one in the right quadrant here, the gray shaded one, formed protein condensates. So always when the Terahertz spectroscopic signature was here, it was a condensate. The other ones stayed here in the diluted phase. So you can test it, and it's a nice way to get it. And I think it's because Oscar, that's why I say Oscar has should go the Oscar for the best supporting actor to water. It's playing a role, and it's tuned, and it's like biology has always evolved in water. So both are important. Okay, give the Oscar to the proteins, but this Oscar to the water. And now, like just as a spotlight on what else you can do, of course, we have shown that we can do it in a very inhomogeneous medium. Can you also do it in real time? I mean, these processes here, which I showed, are very slow. Protein condensation form, and they sink down. So that's on the time scale of, let's say, several hours. That you can do with ATR techniques, you measure and record spectra. Can you do it even more time resolved? Because now we have the correlation, optical pump terahertz probe. And yes, we can. So what we built up a broadband terahertz generation with plasma filaments. You have to go, we went with a sample in a liquid jack to get rid of the windows. So now comes all the technical stuff. So we went in order to do this. So you want to do an optical excitation as a trigger pulse and then follow the reaction in time. So then you have to have a very stable liquid jet. Ours is stable for 2 days. it's in a temperature-controlled electromagnetic shielded room and running there for hours. You integrate and you get here the sensitivity range is 60 dB here. So we will first initiate with the optical pump and the terahertz probe. And here I will only show you, like highlight some experiments where we showed that we can do time resolved terahertz spectroscopy. So here you see now from here in this axis, this would be the terahertz spectrum at a certain time. And plotted here is the time delay compared to the optical pump. So you see here at around 0, we see an overlap of the terahertz and the optical pump, which messes up a little bit everything. So, but after 200 femtoseconds, we can take that as real spectrum. And what we see is we hit just water with an optical 800 nanometer pump, high power, and we create a solvated electron. So you go in, you create this solvated electron, and this is, you have to detect the signal because there are only 10 to minus 4 electron and 10 to 6 water, so most of this will be water. We plot here only the changes compared to before. That is why this here is taken as 0, you plot the changes. You see, you hit it, you first polarize the whole water, and this is this red here. And then we see something staying there over a very long time, so 120 picoseconds. And what is this? So what you do is, do I have this? No, I don't have, no. So what you have is, you can also plot the terahertz spectra here as a function of time, and you see initially it has a center frequency here, and then it goes to another frequency there. We are still struggling with many of the interpretations. So for the solvated electron, we know that we can see the cage formed around it, which is very loosely bound. It's less bound than other ones, than the bulk water. This was done with theory together. So you see a stabilized cavity. And you can also understand it has a maximum around here because it's weaker bound. It was predicted and found that it's weaker bound, the cavity around the solvated electron than bulk water. So that's why it goes in the low frequency range, similar as our 2D network. So we can also do it for other molecules. We see that we start something like a solvent quake, which spreads out. After this, we can follow this quake, and this you can find more in detail in these publications, because I wanted to make one topic clear and not go into the details of these newer papers here. But we can also put in myoglobin. We also see changes here. We see different time scales. We can follow the flow of the energy from inside the protein until it goes in the solvent. And it takes about 10 picoseconds for myoglobin before first it's internally in the protein distributed, and after 10 picoseconds, we start to see it in the solvent. Because terahertz probes the solvent. I always say, if you want to ask the protein, go to the infrared. If you want to ask the water what it does, go to the terahertz. We are much more sensitive to this. And just only as a initial, so what do we do if we excite here with a photon? What happens then? If you have a photo exciter, you will have an absorption. Usually it's known that you go down non-radiative. Here you release energy inside in the protein or in some probe. And then the fluorescence will be shifted in frequency. So you have lost energy before before you have your fluorescence, which you see. So usually if you have like a photo acid, which is then releasing energy by fluorescence, or if you have, and I show you this here, if you have something like a biosensor, which is emitting radiation after photoexcitation, then you see that you always have an energy shift. You have to have a higher energy for absorption, and the fluorescence will be with lower energy. So what happens? Where is the energy going in between? They say non-radiative decay. Well, it can couple to the solvent. And we did that explicitly for the case of carbon nanotubes, which are used as biosensors. So there is this carbon nanotube here. They have an exciton inside. And if you bind it, like with the dopamine, the fluorescence can change dramatically. You see the fluorescence here in black before binding with dopamine and then upon binding with dopamine. This is used in biology as a biosensor for dopamine detection. But what can we do? We can say they didn't know why this is changed so much by the dopamine. You see it, empirically use it, and the question is, what is changed? And our suspect was that what is changed is the coupling, of course, to the water. The hydration water is changed by this. And these changes affect the coupling between the internal energy and the hydration water. And you can couple by hydrophilic water populations. And so what we did is we investigated this. And it was known, you see here on the left-hand side, and this is a study from end of last year. So in the carbon nanotube itself was known to have a spectrum like this. So the excitons here can have a low frequency spectrum, which peaks below 1 terahertz. Water has a spectrum, as you know, this is like 10 wave numbers, so the peak here is 3 terahertz. But there's an overlap in spectra between the exciton and the water. If you have an overlap of spectra, it means they can couple. So you can couple the water and the exciton. And what we could show is, and I just want to do it very short, what we could show is, just look at the right plot, that the fluorescence yield of these DNA carbon nanotubes is linear correlated with the changes in delta alpha which we see. So this is the changes in delta alpha. This probes hydration water. This probes the fluorescent. And what we did see, it's inversely correlated. So the higher the delta alpha is, which probes how good the carbon nanotubes couple to the water, the less is the fluorescence. And just, you can come up with a picture like this. So if you excite a carbon nanotube or biosensor or a photochromophore, then you have an energy transfer here to an excited state. Then this can fluoresce here. But the more you can release, the better you can release the energy here before it fluoresces, the less will be the fluorescence. You have 2 ways to get rid of your energy. Either it goes inside and sticks here and fluorescence will appear after a nanosecond, or before, it can already transfer a lot of energy to the water. And these are then lost for fluorescence. So basically, we could say the sensitivity of these DNA nanotubes is depending on how they influence, how dopamine or riboflavin influence the hydration water. how they influence the coupling of the carbon nanotube to the water. So this was like a new view of how this happens on something which is very biorelevant, this biosensors. And I think it's exciting to see that Terahertz opens this window. So it opens new perspective from the perspective of the water, which cannot be probed sensitively by other ones. Others are very good, fluorescence has very good infrared and so on. They are very good methods. They are very good to probe protein interaction. I think terahertz is like the thing to look at the other side of the perspective, which is very important. So this is just a sketch of the excitons coupling to the water. You see, this was like our artistic view of how this is, but you can read more in this publication. And with that, I just want to summarize and say we have like a new method to do calorimetry by measuring the low frequency spectrum. This opens new perspective on calorimetry, how you can probe the solvent entropy and enthalpy, which I think is very important in biology because this is easily tunable with temperature. It's a weak interaction. You have the strong interactions which you don't change by temperature. These are weak interactions. They are in the range which can be tuned within several degrees, and they are almost zero. You need an almost zero process to be able to tune a process. So that's what we can determine, which has been neglected so far. And with that, I want to thank everybody here. I also say there will be just accepted an upcoming Nature Review Chemistry where we have summarized the idea of terahertz calorimetry in one review, which should come out like I have just the proofs here. So if you want to read more, but if you want to ask more, you are very welcome. I want to thank all the people here. This is our Center of Molecular Spectroscopy, where we carry out this experiment. These are the people, so the great group of people who contributed to this. And here you see the different funding agency. I thank them for their fantastic experiments, and I thank you for your attention.
Thank you very much. A really excellent talk, very, I would say, explanatory and also scientifically very intriguing. I'll give now space if anyone from the audience, we have up to 20 people here, would like to ask something. Just raise your hand and I can unmute you, or just you can write your questions to the chat or Q&A. Meanwhile, I'll take my chance because I have several notes I would like to ask about. So I want me to start with one technical. In this part of the solvated electron, you mentioned that the concentration of the electrons is 10 to minus 4 over 10 to 6 to the 6 of water. So that's, of course, a very low concentration. I'm wondering, what's the scaling of the absorption there so that you can actually detect the differences in absorption, because it's very small effective concentration.
Yeah, our range is 60 dB. The dynamic range is 60 dB.
Yeah. But still, I was wondering if just, well, the differences in absorption, I remember there was 10 to minus 3, which would be corresponding to that. But still, I'm wondering, like, how that small concentration of isolated electrons
for the
transforms
ions, yeah,
enough
we
the
have
solvent
seen for
to have
ions,
the signal.
We have seen 100% change in absorption. So for short species, so it depends how big the change is. So you have this, and of course we had to average over like 2 hours.
I see. Okay,
there was 2 days. Okay. 2 days.
Oh, okay. Then it explains a lot. Of course, it's not a single spectrum. You have to average a lot to have something like...
This is the average spectrum over 2 days, and then you repeat it. We also did, I left this out, you generate more solvated electrons by adding sodium iodide, and you measure if this is like the difference. Then you do an electron capture and you measure the difference with and without this solvated electron capture.
How is it done? Is it what's the trapping mechanism? I'm just curious.
Yeah, you can add the capture of the solvated, and sodium iodide is well known to generate. If you hit it with 800 nanometer, it's generating a lot of solvated electrons.
Okay, I see. Okay, great. So maybe more for proteins.
So you take all these spectra and then this is why, this is like a longer story. So then you take the differences of this.
Okay, so for the proteins, which are also of our major interest, so I really like this story of the condensates and how the different types of water contribute to the interactions and how you can probe that. That's really interesting and makes a lot of sense. I'm wondering if that's why I was asking about different types of proteins, because I seen that in that slide you've seen alpha elastin and FUEC, FUS. What's that FUS? Do you know by heart? What's the protein?
Yeah, this was FUS. Yeah.
I don't know the protein. Do you know what is it? I'm just not important, just curious if you know by heart what kind of protein is that, like how much different it is from elastin, alpha elastin.
Oh, it's very different. So it's one of these, FUS is discussed as one of these which really are forming this Alzheimer neurotoxic.
Okay,
so that is the alpha elastin is more like the one you can handle yourself and buy, and the other one, you need somebody who grows it for you. But this is the one where the medical people are very interested in.
I see. So there I was curious that this is of course very powerful in terms of determining the these variety of these thermodynamic measures. But what about, I know, could you say more about how it relates to the protein structure and dynamics, like when you see these differences in the delta S and the absorption, like can you come out structural, have structural, let's say, interpretation for this apart from thermodynamic?
The FUS is also known to have like one side chain which is intrinsically disordered.
So it's probably very charged, right?
Yeah. So, and this one has hydrophobic patches.
Okay.
And it was like a kind of, they found very often that these protein condensate forming thing have these intrinsically disordered parts. And to me, it makes sense because then you have water which can be released.
Okay. So it's intrinsically disordered,
so
but
they
usually...
were like structurally, they were like not. People thought like this, but then they make sense.
Okay. Interesting. And it's kind of related to, I had a related question to that. So we mentioned there also the slopes in the slide you are showing. So
here, this one here and this one here. That's what we call the
correct.
slope.
So of course it's not like a single slope. You have to find a frequency range where to make it fit, right? So still, but the slope, if I understand correctly, is it steeper for elastin? Looks like that, right? So how does this relate to the protein, let's say, structural? And I don't mean the structural properties in terms of the secondary structure. I mean really in terms of its let's say also its sequence in terms of the charge and so on. Like, is it somehow related to? Can you somehow backtrack from this information also the properties of the proteins, apart from, I know it's of course related to interaction between the protein and solvent, but
it's a difference. So this means a lot of hydrophilic water is retained. So it's always a difference to the dilute, right? We always measure difference. And it means it's very hydrophilic. So it will retain the water.
So it means there are a lot of...
the FUS and some other ones which we have, we also showed another one which you can change, and then the slope goes to 0. When the slope goes to 0 by some mutation, what happens then, and we did see this also in this fluorescence picture, they cannot retain the water anymore. Then the solid aggregates form.
That's interesting. And was it like a single mutation? Because I would be surprised if it's a single mutation, we would say, change it that much, probably like, something.
Yeah, but you can destroy hydrophobic patches because you need a certain area to have this hydrophobic layer. This is also known, like Amish Patel and so the ones are calculating this. You need a certain size for hydrophobic patch. And if you put a hydrophilic protein inside, you destroy this. So here you see this is like what they call N1. This is protein condensates more. And here in the picture, you see more of these strange structures. These are solid aggregates. And we could see the difference in the spectrum. These here have a positive slope, so they retain water. These mutations here releases all water. And then so the slope is 0. This means no hydrophilic hydration, because the proteins are more, the ones which are on the protein crystals, they are more protein on the crystal. And if you don't change the hydrophilic water contribution, it means you have much more protein, but you have the same amount of hydrophilic water. So which means you lost a lot of water.
I see.
If you have 1,000 times more proteins and still the same number of hydrophilic water, it means you lost a lot of them.
Right. That makes sense.
And then it looks solid, which I also find very interesting.
Yeah, it makes sense, right? Because it's simply has less water there, so it has to be more solid in principle.
Yes, in the end, it all made sense. So it was like really, but the spectra are really clear. And
yeah, that's
also, I was
really
surprised to see how simple they are. You could have thought there's a mess when we measured. But you see here, there I can show you. I mean, this, can you see my arrows? This peak here,
I do.
this is a protein. There's a positive here because this will increase if you increase the concentration.
Okay.
But it's much smaller. You know, it's also, you can always see them here. You see it for FAS. That's here, because this will increase when you increase the FAS concentration.
Yeah.
And they are large amplitude intramolecular mode, so they are not so broad. And that's why we take the slope here. I mean, we will not take that point. We will put it from here to here. So you can, these spectra are much broader, the water spectrum. So you can clearly see the differences.
I see. Yeah, that's very cool. That's really cool. I like it. So, well, I also like this Oscar analogy, but if the slide you show in the beginning, maybe if you could update it so it's not the crowd, but it's really the co-actors, right? Which should be getting the Oscars.
Yeah, that's what I say. This is not the crowd. It's the best supporting actor. Yeah.
Yeah, that's. I think really it's change of the perspective in how people think of how the biology works or proteins work. Okay, fantastic. So I don't see any questions now from the crowd here, but I would definitely give Oscar to you, Martina, for the great talk and really inspiring work. So I would like to double check before we conclude. Are you okay as we had this to have this uploaded to our YouTube channel so people can also enjoy it afterwards?
Yeah.
Fantastic. We also do it. We usually do it with including these question and answer sessions. Good. So thank you very much. I'm really looking forward to the reviews mentioned and also this Nature Chemistry paper which was under review mentioned.
Yeah, it's accepted. So as I say, it's the proofs. I have to submit the proofs and then it will be out. So
excellent. So thank you again for your time and definitely looking forward for the next results from your lab and insights. So have a good day again and thank you very much.
Okay, thank you. And I hope that more people will do this because, I mean, it's an open field. You can put in anything, and it will be the first time that they look at the solvent. So
Yeah, definitely. I think it's very important to spread these techniques.
yeah.
Okay.
Okay.
So
Good.
have a good
Thanks
day.
a lot.
Thanks a lot again. Bye bye. Thank you,
Bye
everyone.
bye.